This webinar provides an overview of the Nima gluten sensor third-party validation study. Nima Labs partnered with the Food Allergy Research and Resource Program, FARRP, to evaluate the performance of the gluten sensor. Today I will be presenting on behalf of Nima Labs. My name is Shireen Yates. I'm the founder and the CEO of Nima. We currently have the Nima gluten sensor on the market, and today we will be joined by Dr. Steve Taylor, who will be presenting on behalf of the Food Allergy Research and Resource Program. Dr. Taylor is the co-founder and co-director of the Food Allergy Research and Resource Program and is a professor with the Department of Food Science and Technology. As an agenda, we are going to be providing an overview of the Nima mission, the product overview, and an overview of the usage of the device. Dr. Taylor will take over then to talk through the FARRP evaluation of Nima, specifically the design study with materials and methodology, and then looking at the Nima test results compared to food prepared at different ppm levels, and then the ELISA kits compared to food at different ppm levels, and then finally conclusions from the FARRP testing. So first, Nima's mission is to enable people to be their healthiest selves by giving them the power to know what's in their food. We do this by creating people-friendly products and sensing technologies that identify individual ingredients in food. We generate and share data that tell people whom and what to trust, and we also champion and support people who care about what they eat. For a product overview, I will walk through all the components of the Nima platform. The platform consists of a one-time use disposable capsule that contains all the chemistry, an electronic sensor, and an app that can be connected via Bluetooth to your Nima sensor, and you can see results on the app for both iOS and Android users. As a user, you can also share what you're testing with a community of other people, and you can also see what others are testing on both restaurants and packaged foods. Nima is a chemistry-based device. It is an antibody-based technology. And how it works is you first fill a disposable one-time use capsule with a pea-sized sample of food. You screw the lid shut on that capsule completely, and then the food is then interacting with chemicals in the capsule. You place the capsule in the sensor and it mixes everything up, and then you also wait until the sensor releases a sample of food to the test strip. And what the sensor does is essentially read the test strip and displays the results. Nima is not a quantitative test. If we detect gluten in the sample, Nima delivers a wheat icon. If gluten was not detected in the sample, a smiley face appears on the device. It's a binary yes or no. Nima is a powerful tool and another tool in your toolkit to supplement what you're already doing today. Nima results are only representative of the sample placed in Nima, and they are not a guarantee that the entire dish or food sample will have the same result of the Nima tested sample. Use of this product should not replace education, medical advice, compliance with food labeling, and adherence to avoidance strategies. Say if you're going to eat it anyways, then we encourage you to let Nima take the first bite. We see thousands of our Nima users using Nima whenever they need that one additional data point. We see lots of usage at restaurants, special events, Nima when people are traveling, and when they're at friends' houses. We see Nima as enabling people to have more confidence when eating out and also validating where they're already feeling safe to eat. It's been a powerful tool for thousands. To learn more about Nima, visit nimasensor.com. And you can check out customer stories, reviews, and also test data. You can also download the Nima app today to see what other people are testing near you and also see what people are testing on packaged foods. And I'll now turn it over to Dr. Taylor.
Hi, this is Steve Taylor with the Food Allergy Research and Resource Program at the University of Nebraska. Our group has had the opportunity to evaluate the Nima sensor for the detection of gluten in a variety of different food products. Today, we'd like to share our results with all of you. In the evaluation of the Nima sensor, we prepared 13 different types of food products. Those types of food products included muffins, breads, pastas, that's pasta noodles, puffed corn snacks, ice cream, meatballs, a vinegar and oil salad dressing, oatmeal, and dark chocolate. For the breads and the pasta noodles, we prepared 3 different types of each of those products because we were curious to know if the formulation or composition of those foods would affect the performance of the Nima device. In each case, we determined the gluten-free status of the basic food using the Neogen Veritox for gliadin R5 ELISA method and confirmed that all of the foods or components or mixes were indeed gluten-free. In all cases, the gluten source was a commercial all-purpose wheat flour, and the gluten level of that flour was determined by the Neogen Veritox for gliadin R5 ELISA. For the determination of gluten in all of these products, at all of the gluten levels, we used the Nima sensor and also 2 commercial ELISA or enzyme-linked immunosorbent assay kits for gluten detection, the Neogen Veritox R5 and the Romer Laboratories AgriQuant G12 ELISA kits. After we obtained gluten-free versions of each of the foods, we prepared and spiked gluten into the various food products at 5, 10, 20, 30, 40, and 100 parts per million gluten in the finished food product as ready to consume. The gluten spikes were prepared by mixing all-purpose wheat flour with a gluten-free baking mix obtained from Bob's Red Mill. The gluten spike levels were done on a weight basis, assuming 100% recovery. We mixed each food thoroughly to obtain homogeneity, and the homogeneity was confirmed before processing and preparation by testing 5 subsamples, and an agreement within 20% was considered acceptable. The foods were processed or prepared in the pilot plants or kitchens of the Food Processing Center at the University of Nebraska. To measure the gluten levels by ELISA, all of the products were ground except for the liquid samples, the salad dressing, and the thawed ice cream. As noted already, we independently determined the gluten levels with 2 different ELISA kits, the Neogen Veritox R5 ELISA and the Romer Laboratories G12 ELISA. In all cases, triplicate extractions were done of each food at each gluten level according to the kit instructions, and duplicate measurements were made on each of those extractions. The gluten concentrations were interpolated from the standard curve using the software supplied by the kit manufacturer. For gluten detection by the Nima device, we used the instructions provided by Nima, the manufacturer of the device. With all foods at all gluten levels, 9 separate determinations were made on each product. We made 6 separate determinations of the food product in its intact form, and then ground the food products and made 3 additional determinations on the ground up product. Now, it's important to note that Nima does not recommend that food samples be ground prior to analysis with the Nima device, but we did that because we were curious if grinding would affect the performance of the Nima device. For the intact food products, small pea-sized pieces of the foods were obtained at typical serving temperatures for the different foods, which would be 60 degrees centigrade for meatballs, pasta, and oatmeal, which are typically served warm, frozen for ice cream, and room temperature for all others. These small pea-sized pieces of foods were introduced into the Nima capsule and processed as indicated in the instructions that come with the Nima device. Some of the samples were also ground, as I've just indicated, and in those cases, pea-sized samples of the ground products were inserted into the capsule and were tested according to the instructions provided with the Nima device. It's probably important to note that pea-sized samples can range in weight, and in the cases of these foods, they ranged in weight from 0.03 grams up to 0.94 grams, and that depended on the density of the product inserted into the capsules. Well, what results did we find? The Nima device performed reasonably well in the detection of gluten residues in all 13 food products at levels above 20 parts per million. Almost 93% of all food samples with gluten at levels equal to or above 20 parts per million were reliably detected with the Nima device. We found a very low rate of false positives, 3.6%. Gluten was also detected in several matrices when the gluten level was less than 20 parts per million, including 27 of 78 subsamples at 5 parts per million and 44 of 78 subsamples at 10 parts per million. With these low levels of gluten, the device worked best in the detection of gluten residues in ice cream and muffins. At 30 parts per million, the device detected gluten in 93.6% of all samples tested, and that's 73 of 78 samples. The only ones where gluten was uniformly detected in all 6 samples were 2 of 6 samples of bread C, 2 of 6 samples of pasta A, and 1 of 6 samples of pasta C. At 20 parts per million gluten, the device detected gluten in 79.5% of all samples. For unknown reasons, the sensor was unable to detect gluten in any of 6 subsamples of one particular type of pasta, but in the other 2 types of pasta, the device was able to detect gluten in 5 of 6 subsamples in each of these cases. The device was also able to detect gluten in all subsamples of the other types of foods, with the exception of bread A, where only 3 of 6 subsamples turned up positive, bread B, where 5 of 6 subsamples came out positive, and the corn puffs, where 3 of 6 samples came up positive. I would note that corn puffs are probably the most highly processed form of food that we assessed. Gluten was detectable with the device at the lower levels of 5 and 10 parts per million, incurred gluten in some food matrices, but not uniformly in all subsamples with all foods. With the ELISA methods, the R5 and G12 ELISA kits, we were able to uniformly detect gluten residues in all 13 food matrices at incurred levels of 10 parts per million or more. The gluten ELISAs yielded quantitative results, as they are designed to do, but failed to detect 20 parts per million more of incurred gluten in food samples with 20 parts per million gluten. Only 6 of 13 samples, all with the G12 ELISA, were detected at levels above 20 parts per million. I would point out that the differences that we see here could be related to a number of factors, as I will explain later. While the G12 ELISA seemed to uniformly detect higher levels of gluten in the various food matrices, the gluten spiking levels were initially determined using the R5 ELISA, and the disparities may be caused by differences in kit standards, calibration of the kits, and other factors associated with kit design. I would point out that most manufacturers of prepackaged foods, gluten-free foods, would not risk making food products, knowingly making food products containing between 5 and 20 parts per million gluten. They would all target the development of food products containing less than 5 parts per million gluten because of the acknowledged differences in gluten detection by quantitative ELISA methods, as we observed in this study. This is perhaps the most important slide that I'm going to show. This slide displays the results obtained with the Nima device on the intact food as prepared. Nima recommends that pea-size samples be taken of the intact food product, and in this case we did that. In the left-hand column, it displays the different levels of gluten that were spiked into the food products at 5, 10, 20, 30, 40, and 100 parts per million gluten. And you can see that the Nima device, the fourth column from the left, worked increasingly better as the levels of gluten increased in the products. At 20 parts per million, as already noted, the Nima device detected gluten in almost 80% of the samples, and that result was heavily influenced by the inability of the device to detect gluten in one of the 3 types of pasta in any of the 6 subsamples. The device worked better at 30 parts per million, detecting almost 94% of all samples, and only one subsample was missed at either 40 or 100 parts per million of detectable gluten. And at 20 parts per million and above, as I've already noted, the Nima device successfully detected gluten in 289 of 312 total samples, which is 92.6%. The ELISA methods and the results obtained qualitatively with the ELISA methods are also displayed on this slide for comparative purposes. Grinding did not make much of a difference in the reliability of the Nima sensor. It worked just as well with the intact food samples as it did with the ground food samples. So the Nima instructions to use the device on the intact food as served to you is perfectly adequate. These are the results obtained with the various food products prepared at incurred level of 20 parts per million gluten. As you can see, the R5 ELISA failed to detect 20 parts per million or more of gluten in any of these samples, but did detect gluten in all of these samples at levels above the limit of quantitation of this kit. The Romer G12 ELISA detected 20 parts per million or more gluten in these samples in some cases, but not others, but again was able to detect gluten in all of these food products at the 20 part per million level of spiking. The results obtained on the gluten-free versions of these products reveal that false positives are not obtained very frequently with these devices. As I already noted, only 3.6% of the samples came out as false positives. That's 4 of 111 total samples. In conclusion, in a few caveats associated with the gluten sensor, in conclusion, we conclude that the portable handheld Nima sensor functions reliably for the detection of gluten level residues at appropriate levels in a wide range of different food products. The food products were deliberately chosen to represent a wide range of products that might be available as gluten-free options. Admittedly, we evaluated this device while carefully adhering to the instructions for its proper use. We made no attempt to evaluate its performance when used improperly. Therefore, we would encourage consumers to follow the use instructions if they want to achieve similarly reliable results. The sample volume taken into the Nima device is a small pea-sized portion of the food. Based upon the results of our experiments, the small sample provided a reliable result when the gluten was well distributed within the tested food. However, the presence of gluten-containing particulates could be missed with this sampling strategy. Because the samples are so small, particles might be missed when they're unevenly distributed, as particulates would be in various food products. Of course, I want to note that the commercial ELISA methods also use relatively small portions of the food, 0.25 gram portions, and particulates could also be missed with those samples based on the small size of the samples taken for those methods. This study has been accepted for publication in a peer-reviewed scientific journal and is pending publication as you look at this webinar. It's going to be published in the Journal of Food Protection, and I would invite you to contact me after its publication if you'd like to obtain a personal copy of the publication, although it should be available for free downloads from the journal.